I'm the founder and general manager of Applied Genomics. Applied Genomics has been doing environmental DNA analysis and developing new techniques for eDNA biodiversity monitoring since 2014. And in this presentation, I'm going to be telling you about a collaboration that we had, a recent collaboration we had with Plymouth Marine Labs, in which we had an opportunity to analyze a number of samples which they'd been collecting over a 12-year time period. And the objectives of the project were to compare eDNA methods to conventional survey methods. We wanted to know about eDNA as suitable for fish phenology and diversity, and how well it did for analyzing trends in communities and population genetics. We had a highly successful project, I would say, in which we were able to demonstrate that eDNA reliably monitors marine fish over time. It's highly accurate for biodiversity monitoring on site, and it was found to outperform conventional methods in both quality and speed. And we were able to demonstrate that it had a significant return on investment. Now, the study site, in case you're not familiar with it, was the Western Channel Observatory, which is off Plymouth, on the south coast of England, at their L4 buoy. And if you're not familiar with it, this is one of the parts of the ocean which is probably one of the most well-studied spots in the ocean. It's the L4 Western Channel Observatory has been going for decades and decades. And so the amount of data that's been collected here is really quite staggering. Plymouth Marine Laboratory has been working at that area for quite a very long time. And about 12 years ago, they had the insight or the foresight to start collecting samples, filtered water samples, purifying those, the eDNA found in those water samples, and essentially storing them, not necessarily knowing what it was they were going to do with them. And so they approached us for analyzing these, specifically looking at fish. And we thought that was a fantastic idea because they had about 12 years worth of samples collected on a pretty much weekly basis. And we had the opportunity to also compare their sampling method with our sampling method. We've developed a large volume automated sampler. It's called the in-depth eDNA sampler. You can see it there on the left. And that's a device where it can be tossed in the ocean, and it automatically filters over a long period of time. So just to put that into perspective, most eDNA samples that are conducted these days are collected as point samples. So essentially somebody throws a bucket in the water or a Niskin bottle, collects a few liters of water, filters that. And what we found is the signal that is received by that eDNA sample is entirely dependent on the direction that the tide was traveling at the time the sample was collected. I like to explain it as in, if you were to go into your backyard and you were to smell barbecue, it doesn't necessarily mean you're having a barbecue. What it means is you have to understand what direction the wind was going in order to be able to know which neighbor to whose door to knock on if you want to invite yourself over for barbecue. In order to be able to overcome the natural variation that's in the marine environment, you need a sampler that's going to sit there and sample over a long period of time, ideally 1 to 2 tidal cycles, to allow that water to flow back and forth over the sampler, and thereby get a very large area that becomes more representative of that geographical spot on the ocean. And while that might seem a very large area to say, well, this is what's happened here, zoom out, it's the ocean, it's really, really big. It's actually not a lot of space in the end. This is what we got out of the DNA as far as DNA recovery. There were 460 samples that were analyzed in total. We analyzed 3 DNA markers targeting marine vertebrates, fish, and elasmobranchs. And on average, we had about 20 times more eDNA recovered from the large volume samples as we did from the small volume samples. And this is it. This is probably the world's longest fish eDNA biodiversity time series, as far as I know. It had an awful lot of assays that went into it, and I'm very proud to say that we had a 100% sample success rate, as in we got data out of every single sample, which is amazing, amazing testament to the guys in the lab. And from those samples, we were able to characterize 213 taxa to the rank of species, which is very cool. Now, I'd like to think that Jackson Pollock would be really proud of this plot. And like all great works of art, if you sit with it for long enough, there are some patterns that actually start to come out at you. And the patterns here that we're actually having a look at are seasonal patterns, where you can see slight differences between winter and summer, autumn and spring. Drilling down into that a little bit, you can see which are the species which are kind of driving those underlying patterns. And drilling down even further, you can see the really trendy species that are in there, which are really showing a strong preference for for seasonality throughout the year. But back to Jackson Pollock, and probably the more interesting stuff is when we split this off into years. Here we can see that there's a clear difference between the early years and the later years in the study. If we consider the blue lines on that plot to be richness, higher richness on the left, lower richness on the right. Drilling down into that, let's just think about the data in terms of the number of species that were recovered in each sampling year. You can see that in the orange is the eDNA, in blue is the catch and count conventional scientific trawl data. Unfortunately, the scientific trawling ran out of funding in 2018, so it's somewhat incomplete. But the eDNA shows a similar trend overall to the catch and count data. And you can see how both data sets start out in the high 60s, even high 70s for eDNA, and trail down over time to become worryingly low towards the end of the data. If we look at the underlying trend, though, in the eDNA data, we can see that the eDNA sort of predicts the decline, perhaps 1 to 2 years ahead of the catch and count data, which I think was pretty cool. And for posterity, we had a look at the landings data, which is the fish species declared landed in each year by small boat fisheries, local small boat fisheries. And this is a flat line, obviously, unsurprisingly, because local fishermen are incentivized to maximize their catch quotas. What we don't see in the landings data, and the landings data are really the data on which a lot of fisheries policy decisions are made. What we don't see is the additional effort that the fishers need to go to in order to be able to meet their quotas. So then to dive into the data a little bit further and look at maybe some functional diversity, we've got the trophic levels here, looking at planktivorous fish in this case. We don't really see a trend in planktivorous fish over the time series, but if we look at the generalist fish, we do see a statistically significant decline in generalist feeders over the time period, and that's also the same of the benthivorous feeders, and also true for the piscivorous feeders. Overall, the winners and losers, 25% of the data of the species that we identified showed a positive trend, but only 1.5% of those data were statistically significant. And 68% of the fish that we identified were found to be in decline. 25% of those were found to be statistically significant. So pretty disappointing result for the fish biodiversity overall. However, we're not here to talk about fish policy. We're here to talk about eDNA methods and all the rest of that to be able to uncover these methods. So if we look at the comparison between eDNA and the scientific trawling, eDNA overall recovered more species than the catch and count scientific trawling. And remembering that these are the small volume samples that were collected by PML over that 12-year period, if you scale that down to the samples that we were able to compare directly between the PML eDNA samples in the TAN and the in-depth large volume samples, obviously the in-depth significantly collects more data, and that's simply a reflection on the differences that you get for being able to collect over the full tidal cycle. And we've seen this repeatedly over multiple validation studies. High volume samples do a much better job at detecting more species, more rare species, as you might expect, really. I mean, it should be statistics 101. You exert more effort and you will find more things. Anybody who's a parent of teenagers will know that. Return on investment wise, we see that large volume samples have a 7-to-1 return on investment based on the amount of data recovered compared to cost. And eDNA methods had a 5-to-1 return on investment compared to the conventional methods of scientific trolling, with the additional benefit that eDNA methods are non-invasive, and so no fish are harmed in the collection of the eDNA data. All of this leads to a what are we going to do with it, with this information? Well, what we're trying to do now is set up a system. The problem that we have here in the UK, and I suspect it might be a similar problem worldwide, is that we don't have a systematic method for monitoring changes in marine biodiversity over time. There are a lot of projects that are very siloed. They're all very much looking for very specific things, and so it's very difficult then to compare these data sets and get any meaningful data out of them. So what we're proposing is to have a network of monitoring stations around the country in collaboration with marine and maritime industry stakeholders, fisheries police, natural capital regulators, academic institutes, all the rest of that, who regularly go out to their local area and are able to just basically pop a sampler in the water, collect the sample once a month, and then have it analyzed. And what we would have then are a network of nodes that are regularly monitored all around the country. And then we would be able to see what are the trends that we're seeing, both in a phenology sense, as in year on year, or what are the larger trends, perhaps due to climate change or other sorts of perturbations that are coming on stream. So audience participation time, if you think it's a really good idea, I'd ask you to please register your interest in this, so that what we're doing is we're canvassing people who are interested, so that we can build up a network of evidence that this A, should be funded for here in the UK, but also B, perhaps you are interested in setting this up in your local area. And if that's the case, then let's see if we can get some collaborators together, get some funding together, and set it up for local area monitoring on a large scale at a systematic level, so that then we can actually start uncovering the real trends that are going on in the marine environment. Now, hopefully that wasn't too quick, and hopefully it all made sense, but just in case it didn't, I'm happy to answer whatever questions you might have.